Hello, my name is Cintron. I go to Fort Myers High School, and for my project, I tested the efficacy of surgical cloth and N95 masks in restricting the permeability of the human coronavirus H-CoV OC43 pathogen in the midst of the COVID-19 pandemic. And the purpose of this project is, of course, we're in the COVID-19 pandemic still. The usage of face masks is a highly debated topic, and these face masks are debated on because people wonder if they can actually restrict the passage of microscopic pathogens such as the novel coronavirus, SARS-CoV-2. And that's why this experiment aims to test on the three most commonly worn face masks and the ones approved by the World Health Organization, which are cloth masks, surgical masks, and N95 respirators. And this will not use the actual novel coronavirus because that would be a BSL-3 pathogen, which BSL-3 is not allowed for high school, but BSL-2 is allowed. So that's why in this experiment, the human coronavirus H-CoV OC43 is used, which shares a similar molecular structure to the novel coronavirus, being in the coronavirus family. And first of all, the hypothesis was that if the surgical cloth and N95 masks are each sprayed by droplets 80 to 120 micrometers from an atomizer containing a solution of the human coronavirus H-CoV OC43 pathogen, then N95 masks will prove to be most effective at restricting the permeability and passage of the pathogen through the mask, because according to the CDC, an N95 respirator forms a seal against the user's face, preventing particle penetration around the edges, which is something that surgical and cloth masks do not offer. And first of all, I had to do background research on the topic because this was something I've never done before in last year's science fairs and everything. And years before that, I've always used Petri dishes, which are very basic and not high tech like this. So this is very new for me. And I learned a lot. First of all, I had to research the molecular structure of this H-CoV OC43 pathogen because I needed to see how similar it was to the novel coronavirus, and it actually is almost identical. It has a spike protein structure. It's very small, like around 10 micrometers on average, and it would work very well as a substitute to the novel coronavirus. So the results should yield similar for either one. So that's why that was used. And of course, I needed to know if the nanospray atomizer, if the particles, the droplets sprayed by it, matched like respiratory droplets. Because 80 to 120 micrometers was for a normal atomizer, but I actually had to make sure about the nanospray atomizer instead of what I said in the original hypothesis. And the nanospray atomizer has around like a 10 micrometer on average spray of droplets, which is actually pretty much the same as respiratory droplets. So this is more accurate actually than using a normal atomizer. So it's a good thing I did that research. And of course, I had to make sure it can survive in water as well, because this 1 millimolar solution of the H-CoV OC43 was in water, and it can survive for days, weeks in water. So that was not an issue since the experiment took a day. So that was definitely good. And I also had to determine how to test for the presence of respiratory viruses. That was by far the hardest part because I never knew how to test for viruses, period. And to do that, I found out about the BioFire Torch machine, which the Cape Coral Hospital thankfully had. And the BioFire Torch 2.1 respiratory panel can detect the presence of any respiratory virus, including H-CoV OC43, which works out amazingly for this experiment especially. So that is why I chose that. And it uses a viral transport medium, by the way, so it works with swabs too. You put the swab in it, so that's why it's perfect for this experiment. It works completely well. And the materials needed, of course, were all the ones shown here, like the human coronavirus pathogen, all that, the test tubes, biosafety cabinet. Oh yeah, everything had to be done in the biosafety cabinet, class 2, as according to the safety protocol. And of course, I had to wear PPE, such as a lab coat, sterile nitrile gloves, and safety goggles for this BSL2 organism. And first of all, the negative control was all negative for the presence of the virus, which was good when it was run through the biofire torch, because it tested to see if the sterile mannequin head without any exposure or anything had virus, and of course no. So that was expected. Positive control was spraying the virus solution in the viral transport media, and of course those came out positive, so that was even better. And with everything going well, I had to actually perform the experiment. So for one trial, all the mannequin heads, the masks are put on each of them, the cloths, surgical, and N95, all sterile, all mannequin heads sterile. Cotton swab is placed behind each one, so then it would catch any virus that goes through, if it does, when it gets sprayed. And of course, then the viral solution is sprayed on each one. And then the swabs are each put in their own viral transport media, and this is repeated for 2 more trials, with the mannequin heads being sterilized each time and the mask being replaced. And with all that said and done, everything was run through the biofire torch system, and of course the results actually came out that none of them had any detection of the H-cov OC43 virus, which is great. And honestly, this serves as evidence that the mask actually works in preventing particle penetration, because with the controls being positive and negative, everything going well, it validates the results. And if the virus was not able to go through, then the mask actually worked. It didn't penetrate. Even though it got wet, it still didn't go through. It wasn't detected on the swab, which was behind the mask. And overall, these are all the run-throughs. It has all the results on them, the run summaries. And furthermore, for future experiments, this newfound method of detecting respiratory viruses using the biofire torch is my favorite part of this experiment, because I looked into something called Thieves Essential Oil, which is said to have antiviral and antimicrobial properties. And using this technique, I can see if it actually works in killing viruses. And furthermore, the main obstacle I faced during the actual experiment procedures was when I accidentally used the biofire torch gastrointestinal panel for a few of the samples, and I didn't realize it. And then when I did, it was a quick fix though, because you don't use the entire VTM sample for the panel. So it was a quick fix, but it was an error and it was noticeable. And overall, that's it. That's my whole project. Thank you so much for listening, and have a great day.